All right. Good morning, everyone. Thank you so much for joining our webinar series with the GBatNet. I'm really excited to introduce our speaker this morning. They are a friend and colleague and fellow bat researcher and PhD candidate at Rutgers University in New Jersey. But Pedro comes to us from Brazil, and he has been studying in the States now for his PhD work, studying biogeography and bats. So he's also one of the GBatNet student representatives, and it has been doing awesome research and is continuing to grow as a bat researcher. So hopefully what he is going to be talking with us about today will potentially help inform other research revolving around bats and biogeography, or maybe just help you think about your study systems a little bit differently. So we are going to be having another webinar hopefully next month, and the date for that will be April 9th, and it will still be at 9 a.m. Eastern Time. So stay tuned on our social media pages. We will be posting when our events are going to be. Otherwise, I will go ahead and pass off the controls to Pedro. So without further ado, um, give it up for Pedro. Thank you so much.
Thanks, Dana, for the words. Thanks for the introduction. Thanks for the people who show up today, and thanks for the people who are actually watching this at YouTube. So without further, let me start, right? Let's talk about biogeography. Before the talk start, just 1 minute about me, right? Who I am, right? My name is Pedro. I'm from Brazil. have a bachelor in biological sciences. I'm also a licensed teacher of natural sciences, right? Both at my home state university. As Dana said, I started my PhD a few years ago here in New Jersey. More recently, I got involved in the American Museum of Natural History for my principal investigator. I'm also one of the student representatives of GBatNet. And interesting fact that I just noticed doing this research is that I'm working with bats for 10 years. March of 2025 marks 10 years that I'm working with bats. So very proud of this landmark, right? And also very proud of talking about a topic that I am obsessed, which is biogeography. So for today, right, we have a quick agenda, right? I'm going to talk about broader things such as what is biogeography, what are the biogeographic areas of interest, what factors shape the biodiversity, right? I'm also going to talk about a little bit what we are doing right now in 2025 in terms of science. I'm also going to talk about bats and islands because it's kind of what I study. And at the end, I'm going to actually show my study, present a study case on the work I'm currently doing at my PhD. So I would like to start this talk by showing to you guys this image, right? Which is basically the vertebrate diversity, is a heat map of the vertebrate diversity on the Earth. And as you can see, right, the first thing you notice is that the diversity is not equally distributed on the Earth, right? You have areas with more diversity, areas with lower diversity, right? And apparently those areas with more diversity seems to be surrounding the equator, right? So we kind of see how life is distributed and we kind of observe the broader pattern. However, let's take one region of interest, let's say South America. If we look at South America and we look to different taxons, such as birds, mammals, and amphibians, we actually notice that they have completely different distributions. They have, in terms of diversity, right? So the diversity, even for vertebrates, is not the same. There are differences, right? And of course, that's true for other biological groups. But my point here is to start provoking you all about the whys, right? So why are we seeing this pattern? Why are we seeing this variation? And what is driving this pattern that we see? So what I basically just said, it's the essence of biogeography, right? So when we study biogeography, we are basically talking about distributions of species, right, and ecosystems in both space and time, right? So trying to answer questions, right, such as how did certain species got in there? Why certain species only live in certain regions and not in the others? And what types of mechanisms actually created or shaped the distributions that we see today, right? So again, there is a lot of questions and discussion and perspectives on this. And before we go deep, there is this quick disclaimer that I would like to talk with you, which is talking a little bit more about evolution and distribution of life on Earth. We must always be careful and adjust our scale, right? Sometimes it's okay if we look narrow, if we look for the details, if we look for smaller patterns, but sometimes we need to go broader, right? And search for major rules and search for other, rule of thumbs, right? So always be careful of that while studying biogeography, right? Because things are always connected and they are connected in different scales. So I'm going to start talking about the broadest scale, the most broader scale that we have in biogeography, right? Which are the terrestrial realms, right? So basically, those are organizations on Earth or how the life is majorly distributed on Earth, right? On these 8 terrestrial regions, right? You have the Arctic regions, which is Nearctic and Palearctic at the north. You also have some tropical regions, Neotropical, Afrotropical, Indo-Malay and Australasian and Oceanic, right? So they encompass tropical and non-tropical regions. And you also have the Antarctic realm at the south, right? So again, of course, that is the broader perspective you can look. So if we go and go one perspective further, right, we are going to talk about biomes, right? And you can start seeing some division, right, in those realms. So you can see that, for instance, the neotropics, they are not homogeneous, right? They have a lot of variation, many types of forest, many types of savannas, right? Many types of biomes composing one realm. And of course, we can go and look even further, even narrow our perspective and look at the bioregions, right? For this particular map, I downloaded this map at this website, ecoregions.appspot.com. If you are listening and you want to explore by yourself, you can just Google it and start playing, right, with the terrestrial bioregions. It's a very interactive site, very nice website. But that is basically the most fine scale separation, right, of the biogeography on the Earth that we have. Okay, guys. But again, if you are listening and by any chance you are interested in working with non-terrestrial life forms, well, the oceans are also divided in realms, right? And again, it's hard to talk about bioregions in the ocean because bioregions are narrow. So we're talking about realms, large scale, that encompass a lot of variation, right? Simply because the ocean is gigantic, right? So if you want to work with the ocean, you can. If you want to work with the earth, you can. And if you want to like also work with the contact between earth and water, right? The ocean and the land, you also can, right? Because the coastal and inshore areas also have their own biogeographic patterns, as you can see. All right, guys. So again, what is the take-home message of these previous slides is that life is distributed on Earth in a non-randomized way, right? There is patterns, there is a whole construction on what we see, right? And of course, looking just for what we have now is not enough. We have to go and look at the past, right, guys? So basically, let's say we have those realms, right? And what is, let's say, the most relevant things for the classification of those realms? We are basically talking about temperature and precipitation, right, guys? So if you do a balance between the amount of precipitation and the temperature, right, you are going to more or less define the provinces, all right, or the bioregions, right? So if it's super hot and wet, it's going to be one, super hot and dry, another one, cold and dry one, cold and wet another one, right? So those kind of like environmental factors are, let's say, the main responsibles for those regions that we see. However, they are not the only thing, right, my friends? Because we can look to, for today, the contemporary patterns. However, it's kind of like looking at an iceberg, right? So if we see the diversity only by what we have today, it's just like looking at the tip of this iceberg. And when we go and look deeper, right, we actually see that those patterns, they are product of many, many, many historical events, right? And I'm talking about like natural history. I'm talking about environmental changes. I'm talking about like random events such as mass extinctions, right? So there's a lot of stuff behind what we see today, and it's important for you that is starting or that is interested in working with biogeography to consider those things, right? Before you dive in deep or, you know, making assumptions or studying things, right? So again, let's discuss a little bit more about the most important events, right, that you want to really take into account when studying biogeography. And I'm going to, of course, start with the OG tectonic plates, right, and continental drift. It's a concept that I'm sure that you all are very familiarized with it at this point. We hear about in high school, in middle school, right, which is basically the tectonic plates moving, right, over the millions of years, uh, creating paths, destroying paths, right, but basically affecting the distributions of species, right? Uh, and of course, it's hard to look back without having evidence. So we can look back to fossils and use the fossils as evidence that, for instance, in the Earth back in the day, there were paths that are no longer existing, right? So we can see this by looking at reptiles, fossils, right? That are in South America and Africa, or other ones that are in Australia and Antarctica, right? So fossils can help us to tell this story. However, other things, other living creatures can also help us on this story, right? So let's take, for instance, the marsupials, right? Metatheria. Very cute animals, right? And they have a distribution that today looks like this, right? So they are in American continent, they are in Oceania, right? But how? How did this pattern of distribution was there? So if you just look to this image, to this map on the left, oh, someone is talking. Sorry. If you just look at this map on the left, it's hard for you to understand, right? How does this pattern was created? However, if you look at the Jurassic period, 160 million years ago, then you start understanding why this distribution makes sense, right? So again, looking back at the tectonic plates and the continental drift can really help us to understand biogeography, and is definitely one key factor that you want to keep an eye open for. Another good example that I particularly love is the Gabi, right? We kindly call it Gabi, which is the Great American Biotic Interchange, right? Which was basically when the two, North America and South America, they got in contact with each other by the Isthmus of Panama. And together with this isthmus, right, there was a gigantic interchange of life, right, guys? So because the path was closed and now it's open, you have lineages going from the north to the south and lineages going from the south to the north, right? So again, a good example of by tectonic plates, let's say, generating some generating pathways, right? But of course, we can also have situations where paths are not created but also destroyed, right? So for instance, when you take this example of the Indian subcontinent, right? The Indian subcontinent was isolated for millions of years, and once the Indian subcontinent touched the Asia, right, it created a passage. So everything from the plate could now go to the continent and vice versa. However, because of the tectonic plates, right, and the Himalayas, right, basically a mountain chain was being created, and this mountain chain also was a barrier to the biogeographic corridors, right? And I'm talking about like literal barriers, like there's a mountain and you can't cross, but I'm also talking about other barriers such as like climatic barriers, right? Because the climate changes from the two sides of the mountains. I'm talking about biological processes such as evolution for high altitudes and so, right? So the continental drift is definitely something that you want to keep an eye open, right, when studying biogeography. And of course, I'm talking about going broader and narrow, right? So let's go as broader as we can, right? So how broad can we go by studying biogeography? So I would touch on this idea, right, which is the Milankovitch cycles. Again, I'm not going to dive deep on this, but basically, this guy Milankovitch, at some point, he noticed that uh, the orbit and the axis of the Earth was not static. was a dynamic, right, interaction, right? So sometimes this was changing, right? Sometimes it was in one way, sometimes on the other. But basically, depending on those changes, we would have differences in the amount of solar radiation on the Earth. And the amount and these changes in the amount of solar radiation on Earth will decrease or increase, right? The amount of ice sheets that we have on Earth. And the amount of ice sheets we have on Earth will, of course, increase or decrease the fluctuation of sea levels, right? So it's not only the land masses moving, right? But it's also like what is happening with the Earth in the space, right? And how this affect sea levels, ice sheets, and so. So again, many natural examples. I decided to bring one from our own species, Homo sapiens, right? So how did humans arrive in America? So they probably arrived in America by walk, right? By this land bridge called Beringia, which was only available because of the ice age and glaciations, right? So of course humans crossed, but many other animals also crossed, right? But again, this is a nice example for you to understand that it's not only about tectonic plates and continental drift, but it's also about like this broader Earth processes. Of course, I'm talking about like a lot of geology, geography, but it's not only about that, right? We have life, and life is always interacting, right, with each other. Life is always taking opportunities. Life is always uh, being a product of their own interactions, right? So, for instance, there's many ways, biological ways of changing the communities or changing the distribution of certain species, right? And here I decided to only select three. There's many more, but I decided to select three. The founder's effect, right? Which it's when a new established population only takes part of the original one, right? As we see, and the new population is it's not a good representative of what the old one is. We also have bottleneck effects, right? When we have a more diverse population, something happens and there is a loss of diversity, right? And of course, we have adaptive radiation, right? Where one common ancestor in a new habitat, right? There's a lot of opportunities, so they speciate, they start radiating into new species, right? And so. So again, my goal is not diving deep into the processes, just make sure for you that is listening that there are more things affecting biogeography, right? That only the geo part, the bio part is also affecting it. And of course, I'm talking about the species, right, as they interact, but I'm also talking about our own species because we do have a participation on the biogeography that we are seeing today, right? And I'm more specifically talking about the megafauna, right? So if you look at just like a few thousand years ago, we used to have a megafauna, right? Animals that were can nowhere be found today, right? I'm talking about like mammoths. I'm talking about other big mammals, right? And most likely those animals and their biogeography was directly affected by us, the humans, right? And we could do this by directly hunting them until the extinction or by destroying the habitats where they exist, right? So again, we kind of like we always were interested in biodiversity, more or less, right? The men in caves, they kind of knew what life was there and where you could find certain species, right? However, they never measured their consequences, right? So they ended up directly affecting those species that they were hunting, right? So again, consider at the bio part, right? The animal or plant interaction with themselves and their interaction with humans, right? Because this is also part of the equation. So again, I'm talking about humans and I'm talking about like early humans. So let's dive superficially, right, on how or when did the biogeography started, right? So again, just quick overview on this, right? Biogeography is a relatively new science, right? There are some observations, right? Early observations from the Greek period, right? However, the science, as we know, started in the 18th century, right? With the first scientific thoughts, right? Then the 19th century with the evolutionary thought, kind of like had to rethink many things, right? In the last century, we had a lot of great innovations, a lot of great names, right? That also help us to build and to reach the place where we are now, which is basically what I want to talk, right? So I don't want to go and talk about the past. I want to talk about what we have right now. So what we are doing with biogeography right now, 2025. So there's a big revolution, right, in terms of technology and how this affects the science we do. And this definitely affected biogeography. For instance, today we have a huge access to online databases, right, my friends? And I highlight in GBIF, but you could do, there's a lot of other databases, right? Which basically can give us a precise latitude and longitude of certain species, right? So we know exactly where those species were collected, and we can then take things such as environmental conditions and so, right? So online databases are definitely a game changer for BioGeo. Other thing that is changing the game is the amount of information that is available online, right? Not only on databases such as GBIF, but institutional databases, right? Such as museum collections, right? And I'm showing here some of the museums, and some of them, or I guess most of them, already have GBIF repositories, but some museums, they only have their own websites, their only online repositories. So be mindful of that if you are interested in doing some biogeo studies, right? There's many ways for searching the information. And of course, once you have this information, right, what you do, you have the latitude and the longitude, so what? So you can start, for instance, working with another tool that we have, 21st century, which is the GIS, right, guys? Geographic Information Systems, right? Which basically allows us to combine the data that we have with other types of data, such as topography, precipitation, land cover, human use, and so, right? And to then allows us to do spatial analysis, right? So again, game changer on what we do on biogeography. And again, I could stay here and talk and talk and talk about like contemporary tools and methods, but I'm just basically going to say three of them more, which are satellites, right? Definitely changing the game and the amount and the speed that the information travels. Uh, Presence of historic data, right? My friends, it's very relevant. Now we know, specifically, we know how were the conditions on Earth 20,000 years ago, 100,000 years ago, right? And with this data, we can, of course, do some biogeo studies. And artificial intelligence, that is kind of like still unexplored, but I'm sure that in the future years, this tool will be incorporated at the biogeo, right? So, but again, this leads us to another topic, right? Which is Pedro. What about the bats, right? You're talking about biogel, that's nice, but what about the bats? So I am a gigantic advocate of bats. I love bats. Uh, and especially for biogeography, bats are great objects of study, right? You can use bats to answer many questions in many ways. And again, I decided to pick only two because our time is short. So the first main reason for you to use bats in biogeography, if you're interested, is that bats are super diverse, right? I just double checked it. Yesterday, there are 1,487 bat species and counting, right? Which represents more or less 20% of all mammal diversity we have. All right, guys. So there's a lot of possible objects of study for you to, answer biogeographic questions. And here, of course, I'm highlighting the Philostomidi bats because they are a good example of adaptive radiation. But again, you could do with so many types of bats, right? So many families. There's a lot of good stuff still to be worked on, right? So consider this, right? That you have a lot of options if you want to do biogeo and bats. And of course, if you don't want to guide yourself through the taxonomy and through the ancestrality of the lineages, you can guide yourself to the geographic areas, right? Because bats are basically everywhere, right? Since they can fly. If you exclude the top and bottom frozen areas, right? Bats are everywhere. And that's mainly because they can fly, they can reach regions where no one else can reach, right? And that is super relevant. Here I just selected one example for you on how incredible those bats are. Uh, so for instance, take a look at this hoary bat, right? The Hawaiian hoary bat. with 40 centimeters of wingspan, he was able to, reach the most isolated archipelago on the planet, right? Super rare event. We don't know if it happened only once or twice, but we definitely know that like this is a great example of how how incredible the dispersal capabilities of a bat is. And again, I would like to explore a little bit more about these movements because also bats, because they fly, they can reach places where other taxons can't, right? And this is good because you can use bats for studying the geographical area if you want, right? So you can guide yourself through the biology of the bats, but you can also use bats to talk about a geographic region, right? And in this particular case, I'm going to talk about islands, okay, my friends? Just can superficially talking about islands because they are very, very relevant for biogeography. And of course, you can say like, okay, you are saying this because Wallace and Darwin, they are like the OGs, right? And they were also studying islands in their own works, right? So that's why islands are important, right, Pedro? And the answer is yes, but not only, right? There are many reasons for why islands are relevant for biogeography. And honestly speaking, I do believe that if you want to start working with biogeography, try to take a look at an island, right? Or to an island system. Because, as I put it here in the presentation, is an isolated environment. The boundaries are very well defined. The conditions are controlled, right? And the ecosystems are usually simplified. Okay. And again, there's a lot of materials. There's a lot of historical material available from like hundreds of years if you are interested, right, in studying islands. Um, so be mindful of that. If you are thinking about a place to start doing biogeo, try the natural laboratories, right? And again, why they are so relevant for biogeo? Because, well, a few years ago, right, a few decades ago, two scientists they basically came up with a clear relationship between area isolation, right, and species composition, which kind of like revolutionated the biogeo. Um, and of course, it's a great example for you on how those physical factors can dictate the biological factors, right, my friends? But again, of course, it's a disclaimer, right? Again, perspectives is not only about physical factors, as we already know, there are biological factors also involved into the biogeography of certain species. Okay, my friends? And again, islands are cool. I don't want to dive super deep on it because there's a lot of things more to talk, but basically, once a species reach an island, it's kind of like sink or evolve. There's basically those two ways, right? Or you thrive or you get extinct, right? There's many ways that life got succeeded on islands, right? And again, sometimes the ways they are not reinvented; they are just there in different taxons, right? So let's say we call this syndromes, right? Island syndromes. So it's very common, for instance, you see birds from islands. Some birds they don't fly anymore, or they start losing the dispersion powers, right? Sometimes you go to an island and the animals there, they are not familiar to predators, so they are kind of naive to predators. Sometimes the creatures on the islands, they become smaller or bigger, right? Depending. So it's very nice how those things, how those processes, they are leveraged in an island. And again, for just we talking about bats, I brought this example, right? The Mystacina tuberculata bat from New Zealand, which because there is only one other bat in this island, I guess it's actually the only mammal. There's only two mammals and two of them are bats. There's a lot of there's a broader omnivore diet, and they are even like semi-terrestrial. They can walk, right? Mainly because of this flightlessness, right? Also, you can see in bats. So very interesting, right? Islands, islands, islands. I'm talking about islands. I can talk about islands for like a long time. However, there is an activity for you all. So I'm gonna ask you for imagine an island. Okay, think about an island. And I am almost sure that everyone listening thought about something like this, right? So Earth surrounded by ocean, can be many, can be one single island, can be an archipelago, can be a small island, can be a broader island, right? But I'm quite sure that you all thought about this when I say island. However, a very, very important disclaimer for biogeography, right? Not only for island biogeography, but for biogeography is that islands are a synonym for isolation, okay, my friends? So any region that is isolated or separated for other regions can be considered an island, right? And I'm bringing other examples of natural island systems that are not necessarily islands or surrounded by water. For instance, mountain chains, right? Mountain chains are technically islands. We call sky islands, right? It's basically the peak, the earth, the land in the peak is isolated from the land from the bottom, right? As you can see here, there is like a geographical barrier, right, from the top to the bottom. The distance is there acting, but there is also the ecological differences, right? You have the top one, you have one biome in the bottom, in the base of the mountain, you can have another biome, right? So you have a clear separation between up and bottom of the mountains, and the pattern that we see over there in the right kind of like resembles island in the ocean, right? When you stop looking at. Again, other systems can be islands, and I'm also, of course, bats. So let's talk about caves, right? A little bit. So caves can be islands because caves are, depending on the cave, right? The system can be super isolated and technically closed, right? Which means that whatever is inside the cave will never leave the cave, right? And vice versa. What else is outside is never going to reach the interior of the cave, right? And again, this can be true uh, for many species, but for bats is interesting because bats can kind of like move across, right, out and in, and they're super important because they bring nutrients from outside, right? So again, if you are interested in biogeography and bats, you can consider in working with caves, right, or cave questions. You can definitely do some questions on it. Another thing that can serve as, an island is forest fragments, right? As we can see over there in the image, super didactic, right? Isolated from the rest. Uh, However, the difference from forest fragments is that, uh, again, it's more dynamic than the ocean. Glaciations take a lot of time for the sea levels to rise and go down, but with forest fragments, it's super dynamic. In a question of a few years, you can actually see some difference. So here I just like highlighting the difference of 20 years, 2 decades, right, in an environment, and how this, of course, affects the biogeography of every single life form in that region, right? So again, if you are interested in biogeography, you can also work with forest patching, with, questions related with that. And why this is relevant, my friends? Because, well, as I said, right, before, our species directly influences other ones. And, well, we are destroying our world faster than we can actually count. So the last topic, right, relevant topic on biogeography is this, which is the biodiversity hotspots, right? That's a super important topic if you are doing biogeography. So those areas that you are seeing on the map are areas with a super duper diversity, but also areas that are being like threatened, and especially talking about habitat loss, right? So if you're not familiar with this map, you might think like, hey, but wait, what about Amazon? Amazon forest is not a hotspot, and the answer is no, because there is still a lot of area that is not, lost. For being a hotspot, you need to lost some area. So all those areas you see, they're kind of like they have enormous biodiversity, but they are also, are passing through these problems, right? So again, if you are thinking about doing some biogeography, those are kind of like hot areas for you to, start looking at it. And you are going to see that many of them are islands. That's why I'm like talking about islands over and over. Right. And the tendency, right, the expectation is that probably more areas are going to be included in the future, unfortunately, right? So again, this is a field that is, there's a lot of things, right, growing. There's a lot of things changing, right? So be careful or be mindful of that if or when you are choosing your own questions. I'm talking about a little bit about own questions. I'm just going to show you guys a study case, right, on the, the dissertation that I'm doing for my PhD, which is basically evolution and biogeography of a widely distributed bat in a tropical island system, right? So let's more or less, right, try to see all those things I said in practice, how we actually to answer the questions, right? So for this, let's go back on time, 2021, when I started my PhD. I was super interested in speciation, right? And more specifically, the drivers of speciation. And of course, I worked with bats for many years, so I was like, what bat species I can use for, understanding better the drivers of speciation. And after a long talk, we decided to choose a species that is both widely distributed and that is happening fragmented landscapes, right? Biogeographically, they are very, this combination is also a good combination for you and your studies if you want to do something biogeographically, right? Broad geographic ranges and fragmented landscapes. So with this in mind, we decided to work with this bat species, Epithescus fuscus, super cute, and widely distributed, right, from Canada to South America, Central America, the Caribbean, right, as you can see in this gray map in the left. However, this map, of course, in real life, there is not uniform, right? It's not homogeneous. It's actually like 50 shades of green. There's a lot of echo regions in this uniform green shade that we see on the left. So this might be an evidence, or this might actually be a driver for speciation on this bat. And of course, when we look at the isolated or the fragmented landscapes, islands, right, we can also see that the islands has bioregions, right, itself. So it's a nice candidate for start looking, right, and see and trying to answer biogeographic questions. So my question of my first chapter in my PhD was basically like, okay, I have this bat, it's super widely distributed. I have the mainland lineages and I have the island lineages. My question was basically like, are the island lineages part of the same group of the mainland ones, or they are part of something else, right? So how to study this in the biogeographic context, right? So again, the talk about scale, it also makes sense here. You don't want to look things only under one perspective. You want to collect perspectives and see, what those evidence tell you. So we started by looking at the phylogenetics, right? The DNA. As you can see here, some phylogenies, some admixture, some coalescence analysis, right? Basically, this is saying us that uh, the island bats are unique compared with the mainland ones, right? They have no mixture, they all coalesce to the same common ancestor. They have a very structured nuclear DNA, right? So genetically, they are something else. However, we also were interested in the morphology to see if the morphology, matches with that pattern that we observed. So we took it like 82 skulls and we measured like 16 craniofacial measurements, right? And we run some analysis, right? And what we found was basically the same thing, right? The Caribbean bats or the island bats are unique in comparison with the mainland ones, right? As you can see here in this LDA and PCA, let me just quickly explain. Each dot you are seeing is a bat, and each bat is that set of 16 characters, right? So what we see here is the morphospace, and we see that in both analyses, right, the morphospace, the bats from Caribbean, they have their own morphospace, right, or they are separated in their morphospace compared with the mainland ones, right? So again, they are different in DNA, they are different in the morphology. What about the niches, right, and their habitats? So for doing this again, I kind of did what I said to you guys. I used the GBIF. I went to the GBIF. I downloaded a lot of records for this bet, but I also went to old books, right, or papers, and I mined manually extra records, right, my friends? The final product was something like this, right? So what we see here in the map, each dot is one bat, and we have more or less 400 bats for this data set, right? So my question is basically like, well, people are people consider those 400 bats as the same thing. We saw that genetically they're not the same, so that morphologically they're not the same. What about the environment? So I'm basically comparing how the groups use their environment comparing with each other, right? And again, what I found was this, those are ENMs, also a product of what we do in biogeography, right? And we basically were testing for habitat suitability, right, guys? So I created three models, A, B, and C, as you can see. Uh, and the model A was created for the Caribbean and projected for the mainland. The B was created in the mainland and projected in the Caribbean. And the C was also created in mainland and projected in the Caribbean. And what we see, right, when I overlap all of them, is that there is a clear pattern, right, a clear difference between the suitable habitats, right, which means that the bats are also using the habitats in a different way. And of course, I did not only did this visually, I did some like quantitative analysis, some stats, right? A lot of nerd stuff that I'm skipping here. But basically, this also showed us in a quantitative way that uh, the niches are different from each other, right, guys? So we kind of like came up with the conclusion that the bats from the islands are different from the bats of the mainland after looking to those three perspectives, right? And we decided to also rename this new group, this new island group, right? Taxonomically, okay? So again, I talked a lot, I'm skipped a lot too. If you're interested in double checking the results of what I did, that's the paper we just published last year. I just checked it today, and it's nice, almost 1,200 downloads, three citations, nice stuff, and you can, go into details on the things I just said. Okay, Pedro, we know island beds are different, thumbs up. But what about the next step, right? So again, we created this group, Epithecus dutertrus, which is basically island beds, right? The island lineages. However, we do understand that the islands are very different from each other, right? If you compare Jamaica with Bahamas, Hispaniola with Puerto Rico, Cuba with Bahamas, right? You see a lot of differences, right? As I said, right? Also related with geography, ecoregions, right? And so. So we know that this group that we named one single group is present in a variety of habitats, right? So considering this, considering that this bat is everywhere in the Caribbean, and considering that islands are a good place for studying evolution and speciation, right? And because they drive speciation faster than other places, I was interested in answering more questions, right? But basically, like, how many groups do I have, right? These island beds are one big group, it's two groups, it's three groups, four groups, five groups, right? So that was what I was interested. So again, I went to some museums this time physically to collect some some skulls. I took it almost 300 skulls, right? Which is like basically 98% of the material available in the US. There was a lot of work involved. I took the same 60 measurements, right, that we did. And I also did some holotype studies. And again, my goal is to understand how many groups do we have, right, guys? So it's kind of like where the artificial intelligence, machine learning part goes, right? Because we have those records of people across the years, like, okay, I captured this bat in this island, so that's the ID. I captured that bat in that island, so that is the ID. But what we are doing right now, 2025, is like removing all the IDs, give only the measurements to the algorithm, and ask the algorithm, are those measurements, how do you cluster those measurements? How do you classify those measurements, knowing that there are no labels, right? So kind of like reviewing everything that was done. So again, like basically uh, showing what we found, right? We analyzed the sexes, right, and we noticed that, of course, for this case, females were larger than males, which again was already observed for Vespertilioniid bats, right? So nothing new here. However, this is where the interesting part starts, right? So in the previous chapter, we named this group as one single unit. However, when we do a PCA and the LDA, we find this pattern, right? A kind of like a cluster, right? among the island groups, right? So if everything was the same, technically there was going to be only one cluster, right? But what we see here are five clusters, okay, guys? So we see that bats from each island, they have their own differences already, right? They already accumulated so many tiny differences that the algorithm can, easily observe for us. But again, here the analysis show the clusters, but I'm going to go deeper, right? And ask and do another analysis to see how many groups those clusters represents. Because we have five clusters, right? But do we have five groups? So that's what kind of the question I was asking myself. And again, I did some really heavy machine learning ideas, right? Using everything I have, right? Not going into much detail here, more into the results. What we found was actually that even though we have five clusters, we have three groups, right? And this is based on normal distributions, right? The statistical support for three groups was the highest, right? And of course, I'm looking to how those groups are distributed, and I kind of see a non-random pattern right here. It seems that the three clusters are organized into the near mainland, Hispaniola and Puerto Rico ones, right? As you can see. And again, so I kind of saw like how many groups exist, right? So then I was like about to explore how those groups are structured, right? So as you can see, right, that's a box plot of each of the 16 characters. And what we see is actually a morphological gradient, right, my friends, from the first, second, and third group, right? Which basically tells us that, as you can see, right, the Puerto Rican ones, the one in the right, the average, the median, right, it's the highest for basically all the measurements, which shows to us that this bat is becoming larger, it's becoming bigger, it's right, the size is literally increasing in comparison to the other ones, right? Uh, so we kind of like noticed this pattern, very interesting. We are still going to publish the results, right? But we kind of like faced this situation where we have the old taxonomy and we have a new taxonomy based on our new analysis. So how to fix, right? What should we do? How we go and we dive deep. So for here, I took the same analysis, the same LD from the previous slides, and we did some holotype tests, right? So if you don't know what a holotype is, it's basically the representative for the species, right? So let's say Epithecus fuscus from Bahamas. There is one bat collected in 1897 that represents all the bats from the Bahamas. And that is true for all the others, right? So we basically took those representatives and we analyzed how good they are actually representing, the group they belong. And what we see is like there's a lot of variation. There's much more variation than we thought it was existing, right? Based only on the holotypes, right? So as you can see, in some groups, like the lineages over here, right? The holotype is overestimating the diversity. So by the holotype, it seems that the group is so much diverse, but it's actually not, right? And here, when the wet Mori, right? Same thing, right? But in this case, it's not overestimated, it's underestimating the diversity of the group, right? Because it's falling on the morphospace closer to this than to the group itself, right? So again, there's a lot of nerdy stuff. There's a lot of like analysis. There's no time for talking about everything, but there are ways for, like not only doing these philosophical, broad biogeographical questions, but also doing some more targeted questions, for targeted groups and targeted systems, right? So again, let me just pass through that slide because the time is getting short. So again, this is not published yet. Uh, the first chapter is, you can always double check what I did. But the second one, the things that I just show you right now, we are still going to publish soon this year, right? And again, this is not the end of story, right, guys? Because more than interpret the patterns of biogeography, we want to reconstruct, right, the evolutionary history and, natural history of the species. Because as I said in the beginning of the talk, those things are very relevant for us to understanding the patterns that we are seeing today, right? And with this, I finish my talk. Thank you so much for you all that show up. Thank you so much for you that is listening on YouTube. Thanks so much for Dana for putting this together, inviting me, right? And for everyone that supported my project. Thank you so much. And that's it.
There we go. Okay, for some reason my camera wasn't wanting to work. Thank you so much, Pedro. That was absolutely stellar talk. I learned a lot. And like it was just an incredible presentation. I'm so happy that you were able to give a talk for us today. Do we have any questions for Pedro before we sign off for the day?
I mean, if you don't have any questions right now, or if you are watching and have any questions, feel free to, send me an email or follow me on Instagram if you want to do this more informally, right? We can always talk science. I love talking science and talk about my work, talking about ideas, collaborations, and so. So thank you so much. I feel like people might not have questions.
Yeah,
And
it looks like we just have people thanking you in the chat. I'm not sure if you have that open, but we got two people saying thank you that you did that. You did an incredible job, and I could not agree more. The research you're doing is absolutely stunning. I got to see Pedro give his presentation at one of the BAT conferences earlier this year, and uh, it was he won an award for a very good reason. But if we don't have any questions for Pedro today, he did put his email and his Instagram handle. They are off to the side there. So if you want to contact Pedro about his work or to nerd out about science, which I can also attest that Pedro is a wonderful person to geek out about science and bats and machine learning with. So please write down his email if you wish to. Otherwise, we can go ahead and log off for today. And if you want to come back yeah, thumbs up. Okay. I will be putting the recording up on YouTube later today. And so you can go back and visit it if there's something you wanted to refresh on. And you can also find Pedro's publications as well that he posted up here, which are also absolutely fascinating and very good work. All right. Okay, well, thank you everyone for tuning in today. Thank you so much, Pedro, for being our speaker today. And I hope everyone has a wonderful rest of their day or rest of their evening or a very good night's sleep. So have a wonderful day, everyone, and stay tuned for our next GBatNet webinar. Bye.